You just heard a beautiful talk from Dr. Hood about how one can study thousands of proteins and get a lot of information from these kinds of analysis. What I'd like to do now is to move on and really speak about one protein and show you how extremely complex even the characteristics of a single protein or a single type of protein can be. This protein is Ig4 antibody. And the reason really why I chose this the subject for this talk is that it really is a really presents a very beautiful illustration as to how academia and industry can work together to really on the one hand solve very nice, very important science questions, but on the other hand then work on developing platforms that have the robustness so that they can be applied at the industrial level and also can generate therapeutic molecules that have the desired developability characteristics that we need to make new therapeutics. Now the first slide shows you a textbook example of what Ig4 antibodies represent. This is from January 2008, where Ig4 antibodies are described as one of the four human Ig4 subclasses that have no complement activation. Ig4 antibodies are supposed to have no affinity, high affinity interactions with Fc receptors, no ADCC, and also that is one of the reasons why these molecules were chosen for the development of therapeutic antibodies that needed to be inert so that they don't activate effector functions, maybe if you would like to use them for certain blocking functions. Well, when do you see Ig4 antibodies? Well, they sort of appear after chronic stimulation with soluble antigen. For example, we see them lots in allergy, chronic parasite infections, but also in HIV. We see them also when people are being treated with the recombinant antibodies we often make after prolonged treatment, Ig4 antibodies. So when we set out to think about using Ig4 antibodies for therapeutic application, we thought we needed to wait a moment and really look at some of the peculiar characteristics of Ig4 antibodies that had been described in previous years. And some of this work was done by Janine Schumann, also here in the audience. So when you look at Ig4 reactivity in sera of patients, allergic patients, then there's some peculiar reactivities. And that's shown on this slide. So here we look at 3 different patients. You see the patients here 1 to 3. And then we have, these are patients that are allergic to either BATV1, which is a birch pollen antigen. This is shown in the assay on the left. Then there are patients that have Ig4 antibodies against FeLD1, which is a major cat dander allergen. It's the assay in the middle. And we also looked in an assay where we cross-link these 2 antigens. So interestingly, in patient number 1 who just have antibodies against the BATV1, we see nice reactivity against that antigen. Patient 2 has reactivity against just FeLD1, we see nice reactivity. But then the third patient has 4 high titers of Ig4 antibodies, both against BATV1, but also against FeLD1. Now this patient on the right, you see, has also bispecific reactivity in the serum. So there are antibodies in there that can cross-link BATV antigen with FeLD antigen. And the hypothesis at that time was that potentially these Ig4 antibodies might be able to exchange half molecules, or Fab-arms as we call it, as you see on the top, and thereby generate bispecific reactivities. And we set out to really answer that question. So the way that was approached was to initially make recombinant antibodies. So here we have an example where we make 2 Ig4 antibodies, one against BATV1, a recombinant against BATV1, and a recombinant antibody against FeLD1. We mix these 2 antibodies and inject them into a mouse. We have a second experiment on the right where we did the same experiment, also added an excess of an irrelevant Ig4 antibody. So a third non-specific reactivity there that could potentially compete with these molecules for activity. And in the bottom, the same antibodies were also expressed at Ig1s. And then we injected them in mice and looked what happened. And what you can see is that we were able to show that bispecific reactivity is generated in mice that are injected with these 2 Ig4 antibodies. And that's shown in the purple. So if you mix Ig4 BATV1 with FeLD1, injected in mice within 24 to 48 hours, we see that about half of the reactivity is bispecific. If you had an excess of an irrelevant Ig4 antibody that is not detected in this assay, in this cross-linking assay, then that competes with this reactivity. So apparently also this excess Ig4, this non-specific Ig4, can participate in this reaction. Then we went on to demonstrate, we also look for the requirements that are required to do this in vitro. This took quite a lot of time. Initially we looked at just combining two Ig4 antibodies, mixing them with cells, and any cell type we added to these reactions led to the generation of bispecific reactivity in vitro. We found out that the important reagent here was a small molecule, and then it turned out to be a reducing agent, which we presume to be reduced glutathione that's present in these cells. So indeed, on the bottom left, you can see that if you take two Ig4 antibodies, in this case these are human antibodies, direct against EGFR receptor that we can code on a plate, and CD20 where we have an anti-idiot type antibody for, so we can detect it. We see that we mix these two antibodies in the presence of reduced glutathione. In green, you see you get nice bispecific reactivity. Again, within 12 to 24 hours, this reaction is complete. About half the molecules are bispecific. The controls, if we don't have a reducing agent or we use Ig1 antibodies, nothing happens. On the right, we show that this is not the result of aggregation, but really a bispecific molecule is created. We show this by mass spectrometry. So on the bottom, you see that if you have the mixture of the two antibodies in the absence of reducing agent, we see that we can find the two molecules back by mass spectrometry with a mass of 1.14515 and 145.9 kilodaltons. If we add glutathione, we see a third reactivity just in the middle, has exactly the mass of the mixture of the two heavy light chain pairs. So they have a new antibody, which is the bispecific antibody with a mass of 145.7 kD. And also in terms of stoichiometry, even though mass spectrometry is not a real nice quantitative measure, we think that we can deduct from this that the stoichiometry is about 1 to 2 to 1, so about half the molecules in an equimolar reaction become bispecific. So at that point, we think we've proven this point. So Ig4 antibodies, we have to think about them differently. They're not regular Ig molecules. These are dynamic molecules that exchange Fab-arms or half molecules in vivo, and thereby they acquire bispecific properties. Now, usually when you look in serum, then Ig4 antibodies will really not be bispecific in such that they do not cross-link antigens. And that's because basically one arm is specific and the other can be just derived from the full polyclonal repertoire of Ig4 molecules. So it's very unlikely that two antigens are sort of encountered at the same time, and therefore cross-linking is quite unlikely. Also, Ig4 and Fab-arm exchange occurs naturally in vivo, as we showed, and it happens in our blood as we sit in this audience. Now, when we published that data in Science, Dennis Burdney and Wilson had a perspective, which is shown here, and they immediately recognized that this also has some implications for therapeutic applications of Ig4 antibodies. So the idea is if an Ig4 antibody, a wild-type Ig4 antibody is developed as therapeutic, and you inject that into a human, then this would engage in Fab-arm exchange with the polyclonal Ig4 repertoire of antibodies available in that blood, and thereby exchange of arms of the therapeutic antibody with the polyclonal Ig4 should occur. So it was indeed indicated that if such antibodies would not be stabilized, that might be possible, then this might have important implications for Ig4 therapy. And there I should say that already in the literature it was known that you could stabilize an Ig4 hinge by certain mutations which would reduce the number of aberrant bands on SDF page gel. And this was a method that had been employed really to prevent that. And the idea was, does this reduction of these bands also have an impact on Fab-arm exchange in vivo? So the questions were, does Fab-arm exchange occur in human with therapeutic antibodies? And also does a mutation in the hinge, there's a mutation of serine at position 2 to 8, prevent Fab-arm exchange? I'll go more into detail later. So we looked at Ig4 antibodies that were approved for therapy, and here's an overview of that. In that point in time, 2007, there were 2 antibodies approved, Ig4 antibodies approved. This was natalizumab, also known as Tisabri, and Mylotarg, which again tussimycin. What we did was really investigate whether these antibodies had a stable hinge, so a mutated hinge, or they had a wild-type Ig4 hinge. This was not known from the literature nor in the inserts that were available for these drugs. So we did a PET mapping and identified sequence, basically the hinge region. And we identified that natalizumab had a wild-type hinge, is a wild-type Ig4 antibody, and Mylotarg had a stabilized hinge. Now then the question is, if we now dig these 2 molecules, does this stabilization, this mutation in the hinge, does that prevent Fab-arm exchange? And if you have the wild-type antibody, does it indeed engage in Fab-arm exchange? So we did an in vivo experiment. Shown on the right, we mixed several molecules, Ig4 molecules against EGFR and CD20, or with natalizumab and a pair of Ig4 against CD20. We also have had some controls where we mixed with Ig1 antibody or mutated the Ig4 antibody. And we took gemtuzumab, so the Mylotarg, which we also mixed with Ig4 CD20, and we looked whether Fab-arm exchange could occur in these mice. We see very quickly that indeed the gemtuzumab, so Mylotarg, did not exchange. In this case, the stabilization of the hinge did prevent Fab-arm exchange in vivo, but natalizumab did exchange in this mouse experiment, indeed confirming that this is a wild-type Ig4 antibody. So then the question is, what happens in humans? So this really gave us an opportunity now to show that also Fab-arm exchange with Ig4 antibodies does occur in human beings. So therefore we worked with Joep Killestein from the Free University of Medical Center in Amsterdam, who is treating patients with multiple sclerosis with natalizumab. And he had sera available of patients prior to treatment and several time points during treatment. And we selected patients where we had at least 3 time points, so 1 before and 2 time points after treatment with natalizumab. Then we set up an assay to detect biospecific antibodies, which of course is not completely straightforward. So what we did is we took cells that have the VLA4 expressed to the antigen that's recognized by natalizumab, and then we detected with an antibody against lambda light chain. Natalizumab is a kappa light chain, so if it would exchange with polyclonal Ig4 from the serum, whereas about 40% is Ig4, is lambda antibodies, then you would expect that you now can detect lambda antibodies with natalizumab VLA4 reactivity. This was indeed the case. If you see here at T0, and you should look at the open circles, the black circles are controls where we competed with excess natalizumab. You see that prior to treatment, there's no biospecific reactivity in this assay, and then after treatment, already at the first time point, most of the patients have biospecific antibodies. So antibodies that bind to VLA4 and also have a lambda light chain attached. In 2 samples, we didn't see reactivity, but for those patients, we had a later time point at 5 months point treatment, and then we did see biospecific reactivity. We don't know at this point why there's some delay in these first patients in generating these biospecific antibodies. So, really as a conclusion then, we can say that Ig4 Fab-arm exchange occurs in humans, as shown in a therapeutic setting using natalizumab. And we also think that even though natalizumab, of course, is a safe drug, there's no reason, why there should be any problems with that because of this reactivity. But we think in the future it will be very difficult to develop antibodies of wild-type Ig4 nature, because this really introduces an unpredictability in the therapy. Because at the start you don't really know with what types of antibodies this therapeutic antibody is going to exchange. And it could generate novel specificities, novel combinations of specificities that are undesired. It's shown that the mutation in the core hinge of Ig4, so the serum at 228 to proline, could prevent Fab-arm exchange, as in Mylotarg. And potentially, we think also mutations in the CH3 domain could be considered. And I'll talk a little bit more about that. And then really we came to the question, so what are now the molecular determinants that that make an Ig4 antibody dynamic. Now there are two major contacts here that are important. One is the hinge region. So the Ig4 has a peculiar hinge in that it differs from an Ig1 hinge, where you have cysteine, two proteins and a cysteine, a very rigid structure. For the Ig4, this structure is much more flexible. You have a cysteine, a protein, a serine, and a protein. And thereby Ig4 antibodies can form both inter-chain heavy chain disulfide bonds and intra-chain heavy chain disulfide bonds. There's an equilibrium between them. Then the other in contact is in the CH3 domains. Also here there could be contacts that are important for the Fab-arm exchange to be allowed. Well, first of all, we showed that the core hinge of Ig4 alone is not sufficient for Fab-arm exchange to occur. So if you take the core hinge of an Ig4, you should see it on the right there. So you have the cysteine, the protein, and the cysteine for the Ig1 in the box, and the cysteine, protein, the serine, and the cysteine for Ig4. So if we put the Ig4 hinge in an Ig1 molecule, we really do not engage this molecule in Fab-arm exchange. And also if we take, so basically that's the same as for an Ig1 molecule. So alone, the core hinge alone is not sufficient for Fab-arm exchange to occur. We think, therefore, we're showing here that both the hinge and the CH3 domain are essential. So what you see here is an Ig4 molecule. Again, we see bispecific activity occur if you expose it to a reducing agent. For Ig1, this doesn't occur. If we take the Ig1 antibody and give it an Ig4-like hinge, there's no Fab-arm exchange. If we take an Ig4 antibody and we give it a CH3 domain of the gamma-1, Ig1, then this interrupts Fab-arm exchange. If we take an Ig1 antibody and we give it a CH3 domain of an Ig4 molecule, so there we do not see Fab-arm exchange. And only if we take an Ig1 molecule and we give it both the Ig4 hinge and the Ig4 CH3 domain, then this molecule now engages in Fab-arm exchange just like the wild-type Ig4 antibody. So both these regions are very important. Now then it's sort of interesting to see what are the amino acids that are important here. So here what we did was we looked at the differences between Ig1 and Ig4. There's a small number of differences. There are also some allotypes Ig1, which allows us to take some of these amino acid changes out of the equation. And then you see that there are really only 4 differences that can be relevant in the CH3 domain. So 355, 49, 419, and 445. We took each of these amino acids and then mutated them individually in both Ig4 antibody and an Ig1 antibody with a permissive hinge. And what you can see here, that really the only amino acid that is relevant is the R at 4,9 in Ig4. If you make that into a lysine, so simple arginine into lysine change in Ig4 interrupts Fab-arm exchange. On the other way around, if the lysine at 4,9, if we make it into an arginine, then that induces Fab-arm exchange in an Ig1 molecule. The arginine and 4,9 are both at the interface between the two CH3 domains. Now, we also looked at other species to really try to investigate further what these requirements of Fab-arm exchange are. So the way we did approach that here, because of course we don't have purified IgG4 molecules from many animals available, we don't even, some of the molecules have not even been cloned. We basically did an exchange reaction where we took 2 human IgG4 molecules and then titrated in serum from a number of various animals. And basically, this is not a perfect assay, but if the IgG4 from that animal can engage in the Fab-arm exchange with the human IgG4, then you would expect that to interrupt that reaction. And that's what we measured. And of course, that's dependent on whether that can occur and also what the concentration of that molecule is and so on. So there are some limitations to this study. But interestingly, we see that for quite a large number of animals, we do see that these can engage in Fab-arm exchange with human IgG4. So we see this for monkey, Rhesus and Cynomolgus monkey. We see it for swine, for horse, for rabbit and rat. So this really suggests that the mechanism of Fab-arm exchange is is conserved in evolution. So we can see that these IgG from other species can participate in the Fab-arm exchange reaction. However, we don't see any sequence consensus when we look at the sequences of these various molecules, at least for the molecules where these sequences were available of. So we believe that the ability to engage in Fab-arm exchange really is regulated by different molecular determinants in these different animals. So in some way, this has evolved in various animals and potentially therefore is an important reaction or important process, but it is not based on the same molecular sequence consensus in those sequences. Now then we did a somewhat close analysis of the Fab-arm exchange in rhesus monkeys. And that was basically that because in our science paper where we first described the Fab-arm exchange reaction, we saw that there we demonstrated that if we give an Ig4 antibody against acetylcholine receptor to a monkey, then this Ig4 can engage in the polyclonal Ig4 with the monkey Ig4 and thereby interrupt cross-linking of a pathogenic Ig1 antibody against acetylcholine receptor. So and thereby the Ig4 could have a protective effect. Really a sort of nagging question after that experiment was that we really did not really know the molecular structure or maybe the molecular sequence of the Ig4 molecules that were present in these monkeys. A number of sequences had been reported. There were some difficulties with the sequences, so this was really, we thought, was like a loose end that we needed to tie up. It took us quite a while to actually solve this question, because initially when we started to clone Ig4 antibodies from monkeys, we found that they were not able to engage in Fab-arm exchange, which was very odd, because we had shown it initially in that first study. Until it turned out that there's a polymorphism in monkey. So when we took rhesus macaques from Indian origin, on the bottom you see that this Ig4 cannot engage in Fab-arm exchange. However, if you take Ig4 from Chinese origin rhesus monkeys, then it does engage in Fab-arm exchange. So there's a difference, a polymorphism there. And luckily in our first study we had used Chinese origin rhesus macaques. Now when we look at the sequences, again that can tell us, give us some more information, and they're aligned here. So here you see alignment of all IgG molecules for humans, from rhesus monkey at the bottom, and then we have the Indian and Chinese origin Ig4 at the far bottom. Now it became immediately clear why the Indian origin Ig4 could not exchange, because it simply has a wild type, an Ig1 type hinge, which we know is not permissive. The Chinese origin Ig4 has a alanine at the third position, so it's a CPAC. Therefore, presumably it's also more flexible and can engage in the activity. Now, if you look at the CH3 position at the CH3 interface, interestingly, the Ig4 from rhesus monkeys does have the lysine at 409, which we know in humans is not permissive for Fab-arm exchange, but there's three other mutations. I at 350, the T at 370, and an L at 405. So that is what I taught you about. So apparently there are other solutions to allow Fab-arm exchange to occur in other animal species. And so the key is in these amino acids. So what is then important in this interface? Well, we believe this is really the interaction strength between the two CH3 domains. So here we used native mass spec to do an analysis of an approximation of the interaction strength between two CH3 domains. At the arrow on the left you see when we look at an Ig4 backbone, and if you have the Ig4 with the R409K mutation, we see that, so this is the mutation that then interrupts Fab-arm exchange. We see that this affinity is in the order of about 10 to the minus 8. If you have wild-type Ig4, this affinity is in the order of 10 to the minus 7. And if the rhesus of Chinese origin, Ig4 is also in that order. Now, if you look at the IgG1 backbone, where we introduced a permissive hinge, we see that the IgG1-CH3 actually have a very high affinity. So it's between 10 to the minus 9 and likely is even a bit higher than that. If we make the IgG1, we give it the Ig4 mutation, K49R, we see that there's about a 50-fold decrease in affinity just by this single mutation of a lysine into an arginine. And if we look at the IgG1-ITL, so the monkey sequence, we see that the affinity is even a bit lower. So we believe that really Fab-arm change requires an optimal KD. If it's too high, the affinity is too high, and this T2CH3 can dissociate. That really will interrupt Fab-arm exchange. If it's too low, then it sort of will fall apart and will not be a good behaving molecule. So really the optimal window is between 10 to the minus 7, 10 to the minus 6. So in conclusion, we have determined the molecular determinants that drive Fab-arm exchange for human Ig4. So this is a proline into serine change at position 228 in the hinge, which allows the formation of intra-heavy chain disulfide bonds for Ig4. And secondly, we need a mutation in CH3, which weakens the CH3-CH3 interaction from a 10 to the minus 9, 10 to the minus 10 molar for the lysine 4,9 to more in the order of 10 to the minus 8 or a bit lower for the arginine at 4,9. Just to show that indeed these are the amino acids that are critical, we just did one more experiment. Here again you see IgG1 molecules don't Fab-arm exchange. IgG4 molecules give nice bispecific activity. If we give the single mutations, we don't allow Fab-arm exchange to occur. And if we have an IgG1 molecule that gets both mutations from Ig4, the protein at 228 and the lysine into arginine at 4,9, then we allow Fab-arm exchange to occur. 2 amino acid changes are sufficient. So just to conclude that, we think that we've shown that the mechanism of Fab-arm exchange is conserved in several mammalian species. The hinge and the CH3 domain are essential. There's no sequence consensus that predicts the Fab-arm exchange to occur. There are different solutions in different animals. We specifically looked at human and rhesus monkey. There are 4,9 and the 228 positions in human, and then the 228 and the 4,5 position in monkey. And I should tell you that ITL, we resolved that actually the F into F4,5L mutation is the one that is responsible for the Fab-arm exchange to occur in rhesus monkey. There's a Fab-arm exchange occurs when there's an optimal KD window, and we think that the conservation of this process really indicates that it may provide a general and immunological evolutionary advantage, potentially by Ig4 antibodies don't cross-link, therefore they can block the activity of cross-linking antibodies that have pathogenic effects. Now that said, we then moved on, as we first indicated, what would be really interesting if we could potentially use this system to make biospecific antibodies for therapeutic approaches. But clearly we cannot use the regular process to make biospecifics, as we already shown for natalizumab, these would engage in the process and would not be stable in vivo. So that's really shown on the left. So that what we set out to do was to design a process now that generates stable biospecifics. And we did this on basis of really moving back to the IgG1 molecule. We know IgG1 molecules are stable in vivo. They have a stable hinge that doesn't allow Fab-arm exchange. And we looked for laboratory conditions that could allow Fab-arm exchange to occur in vitro, not in vivo. And second, we looked for matching CH3 mutations that would favor heterodimerization, so really then to increase the yield of the biospecific antibodies when we did an in vitro exchange reaction. So why did we believe that is possible? Well, that really was kind of a quite interesting experiment that we did with the rhesus Ig4 that I described before. Because interestingly, when we mixed rhesus Ig4 with human Ig4, so we did a cross-species exchange, we could see that we got a much higher yield of biospecific reactivity than when we just did the interspecies cross. I see that on the top. So if we mix 2 Ig4 antibodies from human, we get about half the reactivity, half the binding activity of when we mix a human Ig4 molecule with one of rhesus. And at the bottom you see some additional controls for that. So this suggests that when you incubate a rhesus Ig4 antibody with a human Ig4 antibody, that we get a heterodimer that is favored over the homodimers. So I'll go into a little bit more detail in the next slides, and what you should remember is that really the assay we use in all the following experiments is that we are mixing 2 parental molecules. Each of the parental molecules has 1 mutation, and this differs. Typically it's the K49R in the blue one, and then that may be different mutations in the red one. And then we look for generation of the bispecific molecules. And we need to look for solutions that would favor heterodimerization. We generated a library of different molecules. So in this case we took the blue one, we kept it the same, so it has the K49R mutation that allows Fab-arm exchange to occur. And then for the red one, we made a library where we made amino acid mutations in a number of the inter CH3 domain amino acids. So it's 407, the L368, F405, we knew from monkey, the lysine at 370, and a D at 399. And we exchanged these amino acids for every natural amino acid except cysteine and protein. And then on the right you see the results. The stippled line basically gives you the reactivity that you will see for when you mix 2 Ig4 antibodies. So that's when half of the molecules have become bispecific. And interestingly, I think this is actually quite stunning, is that there are many, many solutions that give you a much better reactivity. So in many of the cases we find that we can find a partner that gives you up to about 100% of the molecules becoming bispecific in this reaction. The one in the middle, in orange, that's the F405L. We like that the most. We also use that in the next experiment, and that was also the amino acid that we found from the monkey experiment. So then after we found this matching partner, we looked for in vitro conditions that would allow us to interrupt or to break a stable Ig1 hinge in vitro, and then we oxidize that thereafter. And what you see here is that we took again a pair of homodimers that have the K49R and the F405L mutation, and then we incubated those with several reducing agents. You see that on the bottom. And we see when we increase the reductant concentration, we see that Fab-arm exchange starts to occur up to about 100% of the molecules engage in that and become a bispecific product. Here we liked the 2 MIAs, 2-mercaptoethylamine the most. It really has the nicest characteristics. At about 50 millimolar, we get about 100% exchange to occur. And we also like this molecule because it's used as a drug in humans. We also presume that we could acquire this at GMP grade for later manufacturing. So this is then the final process that was designed. So you have 2 mutations, the CH3, the K49R in the one parent, F405L in the other parent, and then we incubate this with 2 MIA and about 50 millimolar. We get bispecific antibody formation. Now just to show that this process is very effective, it's almost unidirectional. We use a number of analysis methods. On the left you see a mass spectrometry. These are unpurified samples. So this is an overlay where we basically started with a mixture of 2 parents in the stipples. You see the molecular weight of those, and after we added the reducing agent, again we did a mass spec, and you see that the bispecific molecule was generated, and almost all of the reactivity it has. All of the product has the mass of the bispecific, so that's almost 100% exchange has occurred. On the right we use cation exchange chromatography as an analytical method, Again, as an overlay, you see several peaks for each antibody, which are the typical heterogeneity you see for an antibody with some acidic and some basic variants in there. The two Perrin molecules in blue and orange are there, and after exchange we see that there is one product which is the black one, and if you look very well you see that there are some very small humps just below the blue, the main peak of the blue and the orange, and that's sort of the contamination with the parental homodimers that's still present, and typically this contamination is less than 5% without further optimization. We also did a very detailed analysis of the Ig1s produced, the Ig1 molecules that you generate, of course, are bispecific, but apart from that, they are completely identical to regular IgG. All disulfides are formed in the correct manner. We really don't see any differences with a regular IgG1 product. And also the structure and the function so are fully retained. Now then we set out to see whether we could use this process at manufacturing scale. So really when we design new methods to make novel formats that have increased potency or improved characteristics, then we really also, at least from the industrial perspective, we would like to have these molecules, have this process, we would like to have that this process can be run at standard manufacturing facilities. So really, we really like it if manufacturing can be based on standard unit operations, for example, for Ig1 molecules, because this is how these factories are designed. And we think of both of the upstream process, so the cell production, but also the downstream process, the purification. And we think that really this process, the dual-body process that generates these biospecific molecules, really is very well suited for such application. So what we did was to separately express two parental Ig1 antibodies, both at 1,000-liter scale, just with in a regular manufacturing facility, with a regular purification, using standard Ig1 downstream processes. And then we did the exchange using disposable technologies at the required scale. And this shows that example. So what we did was to, you see here that the manufacturing process at the bottom, where we manufactured at 25-liter scale. We basically mixed the two homodimers, 250 grams each at 25-liter scale. Let it circulate for 5 hours in the presence of reducing agent 2-MeA, and then we remove the reducing agent by diafiltration, which then basically by removing the reducing agent, you get reoxidation of all the disulfide bonds, basically by dissolved oxygen that's then incurred again in the medium. But the nice thing was that, of course, we didn't do this in one step. We used several steps. First, we went from the laboratory scale, so less than 1 gram per liter, 3 mil, to 20 gram per liter at 3 mil scale. You see that at the second graph on the right. Then we went to 1 gram per liter at 25-liter scale. And finally, we did a 20 gram per liter manufacturing at 25-liter scale, generating 0.5 kilogram of biospecific antibody in one manufacturing run. And the nice thing is that really, when you look at the quality of these products in terms of the biospecific product generated, that is actually very, very, very similar. And in each of the cases, we also obtain about 95% of biospecific product. So interestingly, we could scale this process up from basically the lab bench to the manufacturing scale without any further modifications. It really shows that it's an extremely robust process. Now these antibodies that are generated, these biospecifics, are, as I said, completely normal IgG1 molecules. We also have effector function. Here we look at complement activation on the left. So we looked at a wild-type IgG1 antibody against CD20, compared it to a bivalent bispecific CD20 antibody where two CD20 antibodies were exchanged, or a monovalent CD20 antibody where we have one CD20 arm and an irrelevant arm. And we see that complement activation is completely retained. Of course, here there's some loss of activity, basically by some loss of avidity by the two to the one arm. For ADCC, we did the same, and basically here we didn't see any differences. So either of the antibody was bivalent or monovalent, really didn't affect the ADCC activity of the CD20 antibody. We have similar results with other cells. So effector function is completely retained in these molecules. Also pharmacokinetics, you would not expect pharmacokinetics to be affected, but we studied this in quite some detail. And to show you, particularly when you look at the bottom scale, it's the simplest to look at. When we look at biospecific reactivity, we see that this reactivity, the half-life of these molecules is completely similar to regular Ig1 molecules. And just to check that really Fab-arm exchange cannot occur in these molecules, we added a very high excess of Ig4 antibodies, and this did not affect the reactivity, really indicating that these are stable molecules in vivo and they have the PK characteristics of regular Ig1 molecules. Now that I'd like to end just a few more minutes to show you some proof of concept studies, because of course nice that we can make these antibodies, but what can you do with it? So I'll just show you two examples, and of course you can think of many others. But one example is where you can use biospecifics to recruit effector cells, for example T cells. And I'll show you an example where we made a biospecific using this DuoBody technology that can cross-link HER2, a tumor antigen, with CD3, which is associated with the T cell receptor and can activate T cells. And also then a second example, which shows you a monovalent approach, where you actually exchange an antibody against c-Met from a panel that we had available with a well-known GP120 antibody, B12, that we use here as an innocuous arm. So now that's basically irrelevant in the reaction that we are studying to make an antibody monovalent. The first, the CD3 example. So here we made a DuoBody that cross-links HER2 with CD3. You see on the left that if we incubate both T cells and tumor cells that have expressed HER2, then we see that when we add the antibody, we get cells, in the quadrant above, quadrant Q2, we see that there's cross-linking of these two cells by the antibody. And on the right, you see that this is concentration dependent. And we also show some controls there. So if you use control antibodies that either have an irrelevant arm or just are the wild-type antibodies, then there's no cross-linking occurs. Now this cross-linking then also gives you a very potent cytotoxicity. So you have a cytotoxicity assay where we have HER2 expressing target cells. We add PBMC, and then we add the biospecific. And the biospecific, the relevant biospecific, is in black. It cross-links CD3 and HER2. We also have a control, which is in white, with again the CD3 with B12. All these antibodies also have an N297Q mutation to interrupt Fc receptor binding, so we don't get any irrelevant, so to speak, T cell activation. So T cell activation that doesn't occur. So T cell activation only occurs after this antibody cross-linked the T cells with the tumor cells and not via an Fc receptor to monocytes. So we get a very specific cross-linking and very potent killing at low picogram per milliliter concentrations of these target cells. Now then, this also can give you a very potent killing in vivo. Here we have an experiment where we basically take mice, we inject them with tumor cells that express HER2 and human PBMC, and then 1 hour later we gave the biospecific antibody or controls, and you can see that there is a very potent killing of the tumors at low concentrations of antibodies. So at 50 microgram per kilogram, we already get a good reduction of tumor size in this model, which is a fairly potent for when you think about application of therapeutic antibodies. Now another application is where we use this technology to make monovalent antibodies. So why would you like to do that? Well, for example, in the application of c-Met antibodies, many c-Met antibodies have been in development, but one of the problems is that c-Met antibodies have been shown to usually cross-link the c-Met antigen, which leads to activation of that antigen. And therefore, these antibodies cannot be used to inhibit c-Met activation. And you see that in this blot, for example, we have a number of antibodies here. So let's say the fourth and the fifth line, we have two c-Met antibodies, which as a regular Ig1 format c-Met antibody 1 and 2, both give phosphorylation of c-Met, as you see in the black band on the top. However, if we make a monovalent antibody of these, when we exchange them with B12, so now they can only bind with one arm, we see that there is no phosphorylation whatsoever. So by making these bivalent antibodies monovalent, we can generate molecules that are non-agonistic or antagonistic, basically, for c-Met. Now, this is also shown in the proliferation assay. So if we have the NCI H4 for one cells are c-Met positive, if we get the c-Met antibody 2, which is agonistic, we see that it induces proliferation of these cells. However, if we take the monovalent antibody, we see that its proliferation is not active. So again, there's no activation of c-Met by this antibody, and it potentially can be used as an antagonist. And this is indeed shown then in this in vivo experiment, where we use the U87 glioblastoma model. This model has an HGF c-Met autocrine loop. So HGF is the ligand for c-Met, and so these cells grow c-Met HGF dependent. Now, if we add the antibody 001, which is an agonist antibody against c-Met, we see that we can even further increase the growth. So if you treat a mice with this antibody, you even get stronger tumor growth. Interestingly, if we take this agonist and now make it monovalent, we see that it becomes the most potent antagonist, and that's in the blue triangles. So we make, from an agonist, we make it a full antagonist, which basically completely inhibits tumor growth in this model, really indicating that, for example, you can also use this technology to make antibodies monovalent, and therefore use them for certain applications, such as inhibition of molecules or receptors like c-Met. So that I'd like to end. So I hope I convinced you that Ig4 antibodies are not normal IgG molecules. They're actually quite special molecules that can acquire biospecific properties in vivo, which have a lot of implications of using IgG4 antibodies, both in thinking about therapeutic application, but also thinking about immunoassays. IgG4 Fab-arm exchange can occur in a therapeutic setting if you use wild-type IgG4 antibodies. For example, we have shown that for natalizumab. We think in the future that you could not develop such antibodies anymore because they're really, there's some unreproducibility, some unexpected things might happen, and that is something we really like to avoid in drug development. The mechanism of Fab-arm exchange is conserved in several mammalian species. Therefore, we think that the Fab-arm exchange might provide a general immunological and evolutionary advantage, which should be studied further. And then I showed you that we leverage this Ig4 biology knowledge to develop a novel platform for the discovery and development of biospecific antibodies in a human IgG1 format. We have also published this method in PNAS beginning of this year, so if you want to use it, you can basically look at the details in that paper and apply these methods. So I'd like to end just acknowledging my collaborators. I mentioned many of them already. So the group at Sanquin, led by Rob Albers and Theo Rispens, Algenomics, University of Utrecht, who helped us with mass spec, the Free University Medical Center, many collaborators at Genmb, particularly Aaron Labrein and Janine Schuurman, and our colleagues at the manufacturing facility in Brooklyn Park, specifically Mike Grammer, who did the manufacturing of our DuoBody molecules at large scale. That I'd like to end and take your question.
Nice talk, Paul. Yeah, I have a question for you. So this is kind of a speculative type question. And that is, so you mentioned all these different species and how some of them have IgG4s that seem to exchange and some that don't, and even within two different kind of monkey species. And so I was kind of wondering, what do you think the evolutionary pressures are? And for instance, if I think the mouse does the exchange, and so if you were to take a mouse and make it transgenic so that it couldn't do the exchange, and I don't know, gave it some chronic viral infection or something to bring up those IgG4s, what would you expect to happen compared to the wild type?
Yeah, so in terms of evolutionary advantage, I think what Ig4 antibodies do is that they are basically functionally monovalent. So I think they can interrupt cross-linking or agonism by other isotypes. For example, in allergy, you could envisage that if you have IgEs that cross-link antigens, if you get strong Ig4 responses, they can potentially interrupt and mitigate that reactivity. So you could see under conditions of chronic antigen stimulation that it might be a benefit to have Ig4 antibodies. For example, we see in conditions where, for example, in beekeepers, we see Ig4 antibodies. When they're stung repeatedly by bees, in the end they do develop these antibodies. We seem to have some mitigating effects. So I think that's maybe the reason for it. I think there's more investigations are necessary to really pinpoint the effects and also what potentially this might, what the effect might in different animal species. We really don't know, how this translates to the different species. So in the mouse, we know that Ig3 antibodies are the ones that exchange. So that's not the Ig4. They are called Ig3. Actually, they are the different numbers in many animals. So you really can't really say what is the Ig4 or what is the molecular exchange in a certain animal species. You first really need to look at the sequence, maybe even do the experiment to show which one has the characteristics of a human Ig4 molecule.
Hi, a quick question. So in these bispecific antibodies you described, did you look at the role of the residue 228? Did you change that residue?
The 228?
Yes, because you mentioned that this residue where in IgG4 it was changed to serine to proline, and it resulted in reduction of the Fab-arm exchange.
Yes.
However, when you made bispecific antibodies, you did not mention about this residue. Was it changed?
That's a good question. So what we did when we set out to make the bispecific antibody process, we moved back to IgG1. So these have a wild type IgG1 hinge. So it's the proline at 228. And really then we looked for laboratory conditions that would allow this hinge to be reduced so that the Fab-arm exchange can occur in vitro. After that we oxidize and basically the disulfide bonds reform, and now you have a stable molecule again.
Okay, great. Thanks.
So going back to Jamie's question, so lots of species have many FCs, but rabbits only have one. So, and you showed that the rabbit would exchange with humans. Did you show that it would exchange with itself? Have you done that?
That's true. Rabbits have, I thought rabbits would have more isotypes. Isn't that the case? They have one, they have one VH, there's one VH, basically one functional VH.
Well, they have only one FC. And so, I'm just wondering whether this is.
We have not really looked in detail at the rabbit. The only thing what we have shown is that if we take serum from rabbit, then we see that there is some inhibition of Fab-arm exchange. And I think it also was shown by in a paper that rabbits do contain molecules that can Fab-arm exchange. But I really don't know what particular subclass that was, to be frank.
Okay, if there's no more questions, then we have a break.